Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory. My name is Xin Tong, and my role as a technical information scientist is to serve the research community with education and practical solutions. JAX Tech Talk is a 15-minute chat that we answer frequently asked questions for anyone who uses research mice and who doing the preclinical research. And for anyone watch live, please use your comment sections of the video to tell us where you are viewing from and any related questions that you have. We will try to answer those live.
And my name is Janine, and I am a senior technical information scientist. This month, our team in TIS has been focusing on a really popular topic, which is Cre-Lox. Last week, Caitlin gave a live webinar reviewing the basics of this elegant system to make tissue-specific modifications, in which the Cre promoter determines the tissue specificity, and then the Lox allele or the Lox strain is that interesting modification you're hoping to introduce. The week before that, on our JAX Tech Talk episode number 41, Dolores and Paige focused on special considerations for neuroscience applications. Xingtong and I are cancer biologists, so today let's talk oncology solutions using advanced Cre-Lox technology. In this episode, we'll compare several oncology models and how you can modify and monitor disease progression using Cre-Lox. So Xingtong, I was on the bench in breast cancer metastasis research and leukemia research. And the one thing that I've learned in oncology is that there are a lot of different model paradigms and a lot of different model platforms. So I kind of break that up into two major categories, the engraftment kind of side, and then the genetic spontaneous model kind of side. I mostly focused on those engraftment models like patient-derived xenografts, cell line xenografts, and syngeneic cell line engraftments. But I didn't really delve into knockouts, transgenics, those genetic models, and of course Cre-Lox models, which are a combination of knockouts and transgenics. But Xingtong, you did. And so what is the broad question that your lab was really interested in, and what was it about the Cre-Lox system that helped you answer those questions?
Yes, we use them a lot. So our lab was focused on the cancer prevention and specifically cancer chemo prevention and chemotherapy. I used multiple different models. An ABC mean, which is a spontaneous model, as you mentioned before, which will develop the polyps in the intestine and colon, is one of the models I use a lot as well. And another models that since my lab focused on lung cancer and the pancreatic cancer, we are using the Cre to induce recombination for both KRAS as an oncogene or the p53 as a tumor suppressor gene. And in this way, we can introduce this lung tumorigenesis and pancreatic tumorigenesis, so that we can look for the interactions in these signaling pathways and also looking for the molecular mechanisms why the tumor metastasis occurred, as well as looking for a potential therapy to prevent the tumorigenesis in both lung as well as in pancreatic tissue.
Yeah, so I can imagine if you are wanting to study a cancer-driving mutation, like I know in lung and pancreatic cancer, KRAS, like G12Ds and G12Cs are pretty common mutations that are found in patients. And this generates a constitutively active signaling cascade that induces cell proliferation. If you're trying to study this in a model system, you probably want them to be restricted to just the right tissue at just the right time. Otherwise, your model could suffer from developmental defects, or you might get very widespread cancer, so you can't really get that kind of clean tissue specificity that you're looking for, and that potentially even a cancer could develop in the breeder mice such that they couldn't carry the mutations forward. So something like Cre-Lox would allow you to restrict to just those tissues of interest. So in Dolores and Paige's Cre-Lox for Neuroscience episode a few weeks ago, they specifically discussed how difficult it was, particularly in neuroscience, to find promoters that had robust as well as very tight restricted expression to specific brain regions or cell types. And actually a number of those neuro-Cre strains had a lot of germline expression, which ends up completely changing their experimental paradigm. So how clean are the Cre drivers for lung and pancreas in your experience?
Yeah, this seems a little bit different for the cancer study. I mean, for lung cancer or pancreas, we may find a better markers that can be expressed restrictly in the lung tissues or in the pancreas tissues so that we can specifically induce a lung tumorigenesis or pancreatic tumorigenesis. And in addition to that, for lung cancer, one of the approaches I normally use is to use adeno-Cre-virus and doing inhalation, or we call intratracheal instillation. And in this way, they can restrict the expression of the adeno-Cre recombinase in the lung tissue and causes the recombination occurs only specifically in the lung. And then for the pancreas model, I would mention that I normally use the KP model, which is a PDX1 Cre that has been crossed with Flux, the KRAS G12D. And the PDX1 promoter is the one, as I mentioned, it is well restricted to express the Cre recombinase, just in the endo-Cre, the exo-Cre, and the ductal tissue of the pancreas. And for this particular model, it well developed the pancreatic intraepithelial neoplasias. We call it PIENS stage 1, 2, 3, that more mimic to the human disease. And only a very small portion of the mice later on developed the adenocarcinoma. We call it the PDAC, and they do not have any noticeable off-target effect. But again, this model is not aggressive. It takes a very longer time for the tumors, or we see the PIENS to develop, and so that I have enough time to monitor the growth of the adenoma stage and to apply in my preventive therapy on them and see an effect without the control mice getting too sick.
Great points. Just wanted to remind any viewers who are watching live to submit any questions in the comments section of our video, and Xingtang and I will try to answer them about cancer and Cre-Lox. So Xingtang, that KP model that you looked at had the KRAS G12D mutation in pancreas, and you mentioned it has a pretty mild phenotype. But you also described that your lab was interested in the interaction between several cancer driver mutations. So another common mutation in cancer is that loss of function of the tumor suppressor P53. And there's a model that's very related to what you described, and it's called KPC, which takes that PDX1 Cre, the KRAS mutation that you just described, and then it layers on an additional tissue-specific knockout of P53 or a lock-stop-LOX mutant P53 that then renders the P53 inactive. So what does that look like, that KPC model? And then, now that it's a more aggressive model, what kind of challenges would that cause you from an experimental standpoint?
Well, the first challenge I want to mention may not relate to Cre. It's the breeding process. You see, we are combining the Cre, the KRAS, the Flux allele, and the P53 knockout allele. It takes multiple rounds of breeding. I mean, at least take more than 1 year for me to breed this strain in the mice before I can get some experimental mice to use. And again, this model, you are right, it's definitely much more aggressive in my hand. We use a constitutive Cre, and we do see that within 8 to 10 weeks, there will be PIENS developed. And within 14 to 16 weeks, we can see the PDAX in most of the mice. And again, this one maybe not that aggressive that you see, that this is still a pretty popularly widely used model that we can still doing experimental for therapy and the prevention. But sometimes, if you are trying to lay in on more mutations, combining more mutant alleles, it will be creating a model that is more aggressive. And in the way, you may even want to use a tamoxifen inducible Cre instead of just a constitutive Cre, and which can allow you to control the initiation of the tumorigenesis after the tamoxifen induction. And again, this model is still aggressive. It still allows you to study the aggressive disease, and again, a tamoxifen-inducible version may give you more time and also allows you to study the disease in the more forgiving of experimental timeline. And one last thing to mention here is, like, as we know, tumor is more like an adult disease. It may not occur since you were born. So to make it more physiological similar to human disease, you may also want to control the initiation of the tumor at a later stage of the mice instead of just having the constitutive Cre expressed since the mice were born.
Yeah, that's kind of the trick with Cre-Lox in general, is that the Cre expression is completely determined by the expression patterns of whatever promoter that it's being driven from. And so if you've got a very early promoter, which might have really great tissue coverage because it's expressed very early on in development, is more likely to reach all those tissues, it might actually be problematic from a modeling perspective, because then you might be developing tumors, as you said, very early, which would not be physiologically relevant. So that ability to control by tamoxifen is another good way to get that good coverage in terms of tissue expression, but then to hold off on the tumor development until more development has occurred. So we talked a bit about the benefits of tissue specificity and the timing of induction. Something that is common in neuroscience Cre is the ability to track cells with fluorescent reporters. And so there's an example of a confetti allele, which is a Cre reporter, and it's a construct that contains 4 different fluorescent molecules, either a green, red, cyan, or yellow fluorescent protein. And the way that that construct is oriented is such that when the Cre recombinase comes in to recombine those LOXP sites, only one of those 4 colors gets expressed in that cell. So that recombination by the Cre and those 4 fluorescent molecules is stochastic. It's going to be random, and it's going to happen independently in each cell type in which that's occurring. And so that's a really useful tool that was originally developed by the Hans Clevers lab to study stem cells in the intestine and perform lineage tracing, such that they could see that they would have clonal expansion of a certain cell type there in development. So Xingtang, what are the applications for these types of reporters, or just reporters in general, in oncology?
I do wish we have used this allele to combine with our KPC model, but we have not. But I do notice that there's a publications that are using the confetti allele with KPC, and they call it KPCX. And the very good advantage of this technique is that it allows you to monitor, to detect the different tumor clones within the organ, within the pancreas, and also you may monitor and track the circulating tumor cells and track down the origin of the metastasis tumor. And in this way, you may even do more single cell techniques from the metastasis and back to the original tumor, doing RNA-seq, maybe deep sequencing. You can comparing those single cells, but in the single cell level to confirm what are the mutations, what are the oncogene second or even third mutations that cause the metastasis occurred within this model. And another way to look at this would be if you have used fluorescence, or maybe even a luciferase reporter, and you have a way to monitor the tumor growth in vivo, and you can track the responses to chemotherapy, to immunotherapy without necropsy and over several time points.
Yeah, to your point about metastasis, since I did most of my studies as cell line xenografts, I was always looking at a single modifier gene, so I knew exactly what I was testing there. So I could, track my cells with GFP, and I knew that any differences I saw between my control and my experimental were because of that gene that I had manipulated. But I can imagine if you are using a genetic model where there might be several interacting genes, you might also see a difference in the metastasis distribution. So you might see them to different organs, for example, maybe brain versus lung. And potentially you could identify different clones that are coming from those different populations, kind of like that seed soil hypothesis, where depends on, is it the soil, is it the site of that metastasis that's drawing those tumors there, or is it something inherent about those cell types that allow them to establish in those tissues? So I think that idea of being able to then isolate them out and do, single cell or maybe clonal analysis on the gene expression could be really valuable. I also really like your point about the luciferase reporter. Again, when I came from a engraftment perspective, everything that I was doing was subcutaneous, and so I could easily just measure the size of the tumor with a pair of calipers, but I lost out on that, in situ kind of like normal, environment of the tumor because I was putting it in this completely artificial location. So something that you can do with those genetic models is getting more to kind of the physiological relevance is to track them with luciferase because, they're internal and you can't measure them and you can't monitor tumor growth. So it's a really good point. Okay, well, I think that's all the time we have for today. Xingtong, do you want to go over the 2 links that we supplied in the video description really quickly?
Yes, so the first link is for an on-demand webinar that we talk about using Cre-Lox for modeling different disease types, and it contains all the detailed information of how the Cre-Lox can help you to monitor different disease types. And for the second one, JAX offers a breeding services. As I mentioned before, for KPC model, it requires a lot of breeding efforts. I mean, how about send it to JAX, let us breeding the mice and generate the experimental mice for you in a timely manner. And in addition to that, we can even offer advanced techniques just to maybe even doing the embryo cryopreservation, and so that we can generate the mice more quickly in a timely manner for your experiment.
Yeah, that's a good point, especially about that KPC. And then you said, oh, I wish I could have put the confetti in there also.
Yeah.
Just thought that's like so many alleles. And the benefit is JAX has all those alleles already, so you wouldn't have to send them to us. All right, so thanks everybody for joining us this week. Our next JAX Tech Talk is called Let's Talk Safety Studies and Graft versus Host Disease. That's going to be live on Tuesday, September 14th. If, however, you follow JAX Tech Talk on LinkedIn, which I encourage you to do so, you'll get notified anytime we have a new episode coming out. And we're really close to reaching 500 followers on our LinkedIn page. So we'd love for you to help us reach that milestone. And whoever ends up being that 500 follower, I'll shout you out at our next episode. If you guys really prefer YouTube, you can also subscribe to our playlist on YouTube, and you can binge watch all 41 episodes to your heart's content. This is Janine saying stay healthy, stay safe, and stay excited about research. Bye, everyone.
Bye-bye.